Hello students, this is Dr. Faizan Mirza. We are discussing how blood glucose regulation is maintained by the two antagonistic hormones insulin and glucagon respectively. So blood glucose is monitored by our pancreas. In the pancreas there are a cluster of cells called islet of Langerhans. The islet of Langerhans are having good blood supply. This is the islet of Langerhans. This is where the pancreatic cells, the alpha cells and the beta cells, they secrete glucagon and insulin respectively. Alpha cells release glucagon and beta cells release insulin. You can see that the beta cells are present in large quantities and dispersed throughout the islet of Langerhans, whereas the alpha cells, which are a bit larger, they are a bit on the periphery of these cluster of cells. Here, these are the blood capillaries. So the blood capillary allow quick monitoring of and prompt monitoring of the blood glucose by these cells, as well as the hormones which are released by the beta cells and the alpha cells. Alpha cell releasing glucagon and beta cell releasing insulin. They enter right away into the blood capillary, so they can be taken to the target tissue. We can recall that the normal blood glucose range is 80 to 120 milligram per deciliter. This is the amount of blood glucose normally. So right after meal, your blood glucose level rises, and the raised blood glucose is actually because of glucose being absorbed and starch being digested in your digestive tract. The raised blood glucose then is sensed by the beta cells of the islet of Langerhans, as well as the sensory neurons which serve the purpose in hypothalamus. So insulin is then released by the pancreas, and this increases the permeability of glucose to the liver cells and the muscles as well as the adipose cells, and glucose is stored as glycogen in those cells. Glucose also turns into glucose phosphate and it gets trapped. Normally we know that these are some general artificial sweeteners. They do not raise your blood glucose if you take these. The diagram just shows to you endocrine and exocrine part of pancreas both. The endocrine part is what secretes insulin and glucagon directly into the blood, and these cells are the ones which release the pancreatic juice into the duct. So we don't deal with these in the endocrine system because these are largely involved in digestion. The regulation of blood glucose is maintained at a certain set point, and the set point is 80 to 120 milligram per deciliter of blood. If the blood glucose gets raised, the cells which get stimulated are alpha cells and beta cells, and in this case you will have less glucagon in blood and more insulin. As a result, more glucose is taken up by these cells, the liver, muscle and fat cells, and they store these glucose, and the blood glucose returns back to the normal value. If blood glucose drops down the set point, again this is a stimulus, negative feedback occurs, and the alpha cells and the beta cells again are stimulated in a different manner. This time the glucagon hormone is more in quantity than the insulin. So increased level of glucagon circulating in blood cause the liver cells and the muscle cells and fat cells to respond to it and release glucose back into the blood. How do we what function actually is insulin doing? How insulin is involved in blood glucose regulation? So we know that insulin can act on liver cells, on muscle cells, as well as on fat cells. So this cell membrane, cell surface membrane, is the membrane on the muscle. So this muscle cell membrane or the membrane present on the muscle fiber will have a receptor for insulin. And this receptor can bind to circulating insulin if it's present in its vicinity. This is the region inside the cell, and inside the cell there are already vesicles stored, which vesicles are already there, which store the GLUT4 glucose transporter. This glucose transporter is actually just the facilitated diffusion channel, which can allow glucose to enter, but this is stored in the cytoplasm right now. This is a phospholipid bilayer. You know the properties of phospholipid bilayer, that it doesn't allow polar molecules to cross. This outside cell, this is the inside the cell. So rather than drawing all the phospholipid molecules here, I'm just representing them with two lines. So this is, you can just picture that this is a phospholipid bilayer. Now, right after the meal, we know that the glucose molecules, they get absorbed in blood. So there is a raised blood glucose, and this is the stimulus, because the raised blood glucose is actually circulating here. And glucose being polar, it cannot enter these cells because the cell membrane is not very permeable to glucose. You know that is a polar molecule, so it can't cross the phospholipid bilayer on its own. In response to the circulating increased glucose molecule, the insulin hormone is released. So insulin level gets raised. Insulin then binds to its receptor present on the cell surface membrane here, Once the receptor occupancy is achieved, the signal is transmitted along inside the cytoplasm. The signal caused this vesicle to start moving towards the cell membrane, and when it starts to move towards the cell membrane, this vesicle, which is carrying the GLUT4 transporter channels, they get incorporated onto the cell membrane. So the vesicle moves towards the cell membrane, these channels get incorporated into the phospholipid bilayer. The phospholipids just slide past each other. We know they are fluid structures, so they can slide past each other, and these channels become incorporated into the membrane. The incorporation of these channels make this cell membrane permeable to glucose. So glucose molecule, which is in high quantity outside and low quantity inside, a diffusion gradient exists. So the moment these channels are made available on the membrane, glucose molecule starts to enter inside the cell. This is all done by facilitated diffusion through the channel proteins. Now, as the glucose molecules enter, we know that the glucose molecule will keep on entering, and the entry of this glucose molecule will cause the glucose outside the cell to decrease in quantity because the glucose keeps going inside the cells. This glucose keeps on entering until the glucose in blood is again back to the normal range of 80 to 120 milligram per deciliter. What happens to the glucose which is present in the cytoplasm? Once it enters, it can very well leave the cell as well because the transport proteins are very well there. How is this prevented? The glucose gets trapped. It gets trapped by being converted into two different molecules. One is the polymer, which is glycogen, and other is a more polar structure and a larger biological molecule, glucose phosphate, as compared to glucose. This is exactly not a polymer, but this is more polar because its glucose already is polar. So once glucose phosphate is made, so it adds even more polarity to an already polar molecule, and this cannot cross the membrane then. This is how your insulin is involved in blood glucose regulation. Now, how is glucagon involved in the same process? How it is carrying out its activity by promoting the glucose to be released from these cells back into the bloodstream once the blood glucose levels drop. So we know that if blood glucose levels drop below 80, more glucagon will be released. So it acts on the fat cells and the liver cells and your muscle cells, and they will start to release glycogen, converting glycogen back to glucose and releasing it into the bloodstream. Well, technically speaking, muscles do not release glycogen; they keep it stored. But yes, fats and liver cells do release glucose after converting it from glycogen. So here I'm just drawing this phospholipid bilayer of the target tissue, which could be a fat cell or a liver cell, to which the glucagon molecule is going to come and attach. Inside the cell, there will be glycogen stored. So these are the phospholipid bilayer. I'm just adding these molecules here. These are the phospholipid tails and the heads, and these are membrane proteins attached. You have already discussed cell signaling pathway previously as well. This could be the liver cell membrane. Inside the cell, ATP is stored. We know the cells have ATP, and these are the enzymes which are already inactive in the cell cytoplasm. Inactive protein kinase enzyme, inactive phosphorylase kinase enzyme, inactive glycogen phosphorylase enzyme. When all these enzymes are inactive inside the cell, glycogen remains stored in the cytoplasm. Now, this is the glucagon receptor. This is the G protein, and this is the enzyme that's going to make the second messenger for the cell. Now glucagon molecule comes. Glucagon, being a polar molecule, cannot penetrate through the cell membrane. It comes and binds to this receptor. The attachment of this glucagon with its receptor triggers the G protein to shuttle and activate this enzyme. The activation of this enzyme causes ATP to be converted to CAMP, cyclic AMP, which is the second messenger. The second messenger is going to activate inactive protein kinase enzyme into active protein kinase enzyme. The active protein kinase enzyme then goes on to activate phosphorylase kinase enzyme. Once the phosphorylase kinase enzyme becomes activated, it's going to activate yet another enzyme called glycogen phosphorylase enzyme. This glycogen phosphorylase enzyme is what can break glycogen back into glucose. So over this course of reaction, eventually glycogen is broken down to glucose, and glucose is released back into the blood until the value rises back between 80 and 120 milligram per deciliter. Since this is the second messenger involving pathway, we know that amplification is going to occur at every step. That's it from my side. Thank you so much.